When Mikkel asked me about participating here and giving the first talk, I mean, I was really thrilled. I think that's really a challenge that I couldn't resist. So what I'm trying to do the next 20 minutes or so is to give you a view into my world. And of course, I have to say something about design, because I have to admit I'm an engineer in background, but I'm also doing a lot of design. And I'll try to give you an idea of this and also how I think about design. And then there is also this challenge of the colors that I didn't get until I had said yes. And then I had to think about what is color. And you would see there's not so much colors on this slide. Actually only the blue one, which is the color of the Copenhagen color in CreativeMornings. And that's because for me, colors is something that carries information. And you would see that through the talk. So it has to have some meaning, but that would be more evident. So what is it I'm doing? Well, I'm working with and designing computers. I think if you can you lower it a little bit? Get to the top. And you're all familiar with the laptops you're using and so on, but I will give you a quite different view into what computers can be. Of course, we are interacting with the computers, and the way that looking into that is that somehow the computer waits for you to, it sends something, you interact with it, it makes some computation, and the result of that is some action that it's giving back to you. So that could be the normal way of working with computers. But I will elaborate on this and try to take this much, much further than what you could dream about. Well, I say I'm working with computers and designing computers. It's not the laptops and things like this, but it's actually what's inside it. So it's the microchips that are inside the chip, inside the computer. And if you look into that, then within those, you have some small, very small components. And again, here are some colors, because that's very important for us. The colors describes different layers in silicon that carries different functions. So basically, this is a transistor. That's the basic function that you have in your computer. And it's nothing more than a switch. You can turn it on and off. So if I put current on the red one, I can have current flowing in the green. And if I don't, I cannot. So you can turn things on and off. That's the basic component in your computer. How can I use this? Well, I can take those and I can put more and more together. And then I can form small functions in there. And I can put even more together to build small components that are doing something a little more sophisticated, yet more together. And it goes like this. So you try to carry more and more of these. And eventually you get to the size where you have the full chip designed. And at the end, it may not be more than this. And this one is one of the computers that we are using. It has 50 million transistors on it. You can do a lot of computation with that. Well, actually, the largest chips today has 20 billion transistors in them. These are really supercomputers, so you can do a lot of processing. And this, of course, you can use for many different things. So what you should take out of this is that all it takes is that we get smaller, faster, and cheaper. And this is something we are seeing in a number of technologies that is going to influence our life in a very, very dramatic way. And this is what I would like to talk about. So one thing is, and that's the first part, so there are two parts in the talk. The first part is trying to look at the design DNA. So when asked about this, I I did a lot of thinking about what is design for me. How do we do this design of the microchips? Now, if you look at this that I said about that, it's actually sort of a flow where you have input. That's the sensing part. You have some computation, and then you have the acting part. This I will use as the model of computation when I take you to some other areas. If I look into this, then the part that goes for the computation, that's where I really do the design, trying to figure out how to do efficient computation. Now this carries, in a simplified way, 3 different ingredients. So it's the technology. So what is the technology that my computation works in? And in the chips, it's silicon. That is the basic technology. Then I have to think about the behavior. So what is the function of it? What should it do? And last, I have the requirements. So what type of things, what are the conditions into which it should work? It could be size, it could be the power, it could be timing and things like this. Now, if I look at these, then basically what we are trying to do in the design process is to bring these 3 together to come up with the best possible solution. And this is how I conceive this way of design. So the design is not the result in itself, it's sort of a process to get to the solution. Now if we take the computer, well, the technology would be my computer. The behavior could be some simple function. Well, it can actually be quite complex. It could be a full program with millions of lines of code that has to execute efficiently on this. And requirements could be something like timing or power that I can spend on it. And then I want to figure out how to come up with the best possible way of doing this. And of course, the behavior, the language, the computer language has to be translated into something the computer is able to understand. And basically, computers understand zeros and ones, and that's all. So you have to translate it into this. Then I can get it onto the computer and have it executing the behavior that I wanted to. Now, for the rest of the talk, I will forget about the requirements. They're extremely important, and they are the ones that are giving us all the challenges to do the things, but just trying to think about technology and behavior and what could that be. So it can be much more than just the software that you're used to thinking of and the computers as the major thing of doing the computation. Now, let's look back again on the design. So basically, I may have an idea of something that I'm going to work on. And this process of getting there is for me first something where I try to explore. So I try to come up with a lot of different ways of looking at things and different ideas. And this process is, to me, sort of art. It's the creativity process. It's where you really have to be a bit crazy. And sometimes it can be difficult in the scientific community because you have to be very serious. You have to make sure that everything you do is scientifically sound and proven. But in the start process, you have to get out of the traditional thinking. You have to really try to turn things upside down, try to go places where people haven't been before. Then when you have done that, then you can get into the process where you try to reduce, try to figure out which of those are actually the right things to do to optimize this solution that you're going to do. And this is what I call the engineering process. This is where you are very systematic and you can measure and you can compare the different things. But these two goes together. And if you cannot get enough variety in the beginning, you also have a very narrow solution. Now, one of the things that may be different in my world from your world is that as an engineer, we want to try to solve the problems and actually get a computer to do what we are doing. So actually reduces our jobs. So we are actually working with algorithms that can do the reduction, figuring out what is the best. And we also have algorithms that can come up with creative solutions. But of course, unfortunately, I don't have time to go into that. Maybe some other time, but I will carry on this. So let's look at computers and computer systems in a more broad term. So the mobile smartphone that you're carrying around is actually a very, very sophisticated computer. It's very powerful, and if you go back 10 years, 15 years, something like this, the power you today have in your phone is actually what required for doing a huge supercomputer at that time. So a lot of computation in there. You're used to, or we are used to thinking about, we can link our mobile phones to the cloud. This is where you have tremendous computing resources to do all sorts of processing. But there's also a world on the other side which connects to what I call the sensor swarm. So that is a lot of very small computers that can measure things in the environment around us, on us, and so on. And this is the part I would like to continue talking about. So let's try to take it into a context and look at healthcare. So clearly I can use computers to monitor what I'm doing. I have one of these Nike Fuel bands that tells me how much I exercise and so on, and I can keep on looking at curves and seeing how well it's done and so on. But if something is wrong, I still have to go to the doctor. If I go to the doctor, maybe he would say that, yeah, let's do a blood sample, and this blood sample goes to a lab. The lab is doing all the chemical processing of that, getting the result out of this. The result comes back to the doctor, and then I can get my results. Now, if you want to try to turn this upside down and thinking about this smaller, faster, cheaper, then we could think of why not take these things, both the doctor and the lab and the chemical lab, and just put it into the mobile phone. That would be nice, because then I would always have this with me and I can do measurements on the spot and get results on the spot. And this is what we typically call a point of care device. And in principle, that would allow you to have a doctor in your pocket. Yeah? Okay, so just wave if it's, we have to put it up. Now, this is not really a new idea in doing this. There is actually this tricorder. It's from one of the early Star Trek movies in '66. They had this nice device where they could go around and you could actually point it to a person and you could get all sorts of medical data and knowing what was wrong. Now, this has actually then been now part of a global competition under the label of X Prize. So they are doing a number of such competitions, and the competition goes that the team that can build such a tricorder wins $10 million. And there are some requirements for that. So it has to be able to diagnose 16 different diseases, and it has to be done by an ordinary person, not a doctor or anything, and the result of it should be at least as good as the doctors could provide. It started out with 100 teams around the world. There are 10 now in the finals, and by the end of the year they are going to nominate the one that is doing the best on doing this. So we are getting there, and that's really very interesting. But if we can get it so small that we can put it in our pocket, then maybe we can make it even smaller. Maybe we can actually put it inside our body. Let's go back here. So I need to explain a little bit to you why it's possible. It's not just science fiction to actually get it to run in your mobile phone. So we have the chip already. This is where we can have all the processing of numbers and so on. But we can also build what I call biochips. So that is small chemical labs, and that I will tell you a little bit about. So if I look into this chemical lab, I get something that looks like what I had before with the transistor. In here it's not a transistor, it's a valve. It's something I can open and close. And it's actually here again with the colors. So the blue is where I have all my chemicals flowing. And the red one is air. And it's in a rubber-like material. So if I get air into this one, it expands. And then it pinches off the other, the blue one. So by controlling the pressure, I can open and close. Very simple, small device again. But if I have enough of those, then I can do quite sophisticated chemical reactions in there. This is an example of a chip designed some 10 years ago. It has 2,500 valves, and you can see here this is 5 millimeters, so it's not so big. And you can get, with the inlets, you can put in reagents, chemicals, and then you can pump them around, mixing, doing different kind of things that you need to do in order to process this. To give you a flavor of what this is an example of a liquid coming into this circular, which is actually a mixing device. So first you put in one, then you can put in this case, the yellow one. And you can see in each of these places you have valves. When you open and closes those quite quickly, you start getting this to circulate and you can mix it. You can put this out, you can store it, you can do something else. So you can do a very, very complex chemical process in this case. You can also work on this in different ways. So this is another technology where you have small electrodes, and by putting power on and off, you can get small droplets to move around. And again, you can get them to mix. And when they have been moved around and mixing, you can split them again. So you can do all the basic components that you need for doing these chemical reactions on a very, very small plate. And that means that if I can get it this small, I may actually, and people have been doing this, put these things into a pill. So now you have both computation and you have the small chemical lab. You swallow the pill, and it makes samples when it goes through your body, and can do a diagnosis and send the result directly out to the doctor. Okay, so now to the last part of the talk. So now I want to shift. So I talked about the design DNA and the way I'm thinking about it into one where we're doing DNA design. So what is the idea here? Well, the idea is to try again with this notion of being smaller, faster, cheaper, looking into what you can do. You can actually build a combination of the two that I showed you, where you have continuous flow of these liquids, and then you can put droplets in there of other things. And you can get them so small that the volume of them is in nanoliters. That is extremely small. It's actually so small that you can capture cells into each of those. So you can have into each of these bubbles a small cell, and then you can see how you can treat whether certain drugs are actually good or not, if they kill the cell or whatever. Now, if you think of this for a moment, that you can actually work with these very, very small things and start looking into what a cell actually is. So in this case, it's a bacteria. And when you look at the functions that are in there, you start seeing a pattern that resembles very much the way that I explained to you computation. So they can sense something from the environment. That could actually be molecules inside the cell or molecules from outside. And then they react on this, some sort of computation, and then they may produce some other materials. Now this looks to me pretty much like a computer. And then you can start again thinking, now if this is the case, is it something that I could program? And if you look at the things that you can sort of like use for sensing, it could be anything like proteins, chemicals, light, pressure, things in the environment that actually impact the way that the cell will work. And the output could be biomaterials, new drugs, biofuel. So there is a huge potential for doing some very, very interesting things there if we are able to control the things. So biochemists are working on this, molecular bio people. But as a computer scientist, I wanted to really look at whether is it possible to try to view this as a computer and then do the stuff what you usually do, program them, change the behavior of them. So if we look into what is the basic ingredients in it, well, it's the DNA. So here it's just unwind. So you have this double helix spining. And it's a number. It only have 4 different base pairs that goes together 2 and 2. And out of this, you can get a lot of information. This is all the information about how the system should behave. And that also goes for our own cells. Now, looking into this, and this you may also have ever heard about, was this human genome project where a large group of scientists tried to figure out whether they could read the full human genome, understanding what the different things are in there. The human genome has something like 3 billion base pairs in there. So it's a huge amount of things that you have to do, but it's actually possible. And how is it possible? Well, you can take your DNA, you can put it into a sequencer, and that one would actually reveal the data, and you get the data in a form where you can actually work on it on a computer and try to understand what the meaning of this is. Now, if you look into this, and then again with this view of smaller, faster, cheaper, then in 2001 they completed the Human Genome Project. It had taken 13 years to sequence the human genome, one human genome. It cost in the order of close to $3 billion to do that. And everybody said, why do this? I mean, this is crazy. First of all, when it started, nobody believed that this would ever be possible because it would take so long. Now, if you look what happened since then, in 2008, 7 years later, you can do it in 6 weeks for $100,000. And today, you can do it in 3 days for $1,000. And it's continuing. So very soon, we will all be able to get our DNA fully sequenced and getting a deeper understanding of how we work, what kind of things we may likely get later in life. But most importantly is that you can start having medicine being something that is really personalized and not just treating a symptom or something like this, but you're treating the symptom that you have and for you exactly. Now, when doing this reading, people started to look at pieces of DNA and taking those out as small parts, characterizing those and making databases out of that. So that means actually you have a large number of components that are fully characterized. Well, as an engineer, if I have a number of components, I can start put them together, I can start building something. And this is exactly what we are doing. If you're looking into what I showed you with the cell, we had these red molecules that were sort of triggering the cell, and you had the green one coming out. You could think of this as a computing program that says that if we have red, then do green. If not, then do nothing, right? So, so this is a basic function. This is sort of like a very simple computer program. Now, with this one in mind, what could I then do? So let's just take a very interesting example. So malaria. Could we use this to fight malaria? So if you know, in Africa, approximately 2,000 kids are dying every day of malaria. That's a huge problem. So why is this a problem? Well, there is actually a cure. So if we get malaria, it's not really a problem, but we can get the medicine for it. The medicine requires some material called artemisinin, and this is extracted from a plant, the Chinese wormroot, and it's something that is very rare, and it's extremely costly to extract this. This is why the medicine is so cheap, so expensive. Now, the group at Berkeley University, led by Jay Kiesling, were looking into this. They are biochemists, and said, well, could we try to get something else to actually produce this? And then, over some years, they developed, by re-engineering a yeast cell, being able to actually create this. What does that mean? It means that you can actually create this drug by the same way that you're brewing beers. And this is something you can do anywhere, and it's not very expensive. So now they made a company that is actually producing this in order to lower the price and get it more accessible, and maybe being able to really help, let's say, people in Africa and other places with this, which cannot afford the expensive medicine. Now, what they are doing is a lot of trial and error in the labs. But why not, if we were able to, and this is where the computer science come in again, if I could write this program and I could make a system that compiles that into the right DNA and then get it into the cell, then maybe I could hopefully avoid doing a lot of experiments in the lab, or at least reduce the amount of experiments from maybe thousands of experiments into a few hundreds, or maybe less than that. What would that require? Well, that would require that I could translate this into the sequence of these letters in the DNA. I put them in a mailbox. And this is, this you can do today. You can, from your computer, if you have this sequence, you can mail it off to a company. This company produces the DNA, the real DNA for you. And they do this by 3D printing. It's a very advanced and very specialized 3D printer that puts together all these base pairs. And then in the post, you get this back, and you can go in the lab, and you can treat it, and you can put it into cells. So you can actually get this whole process working. What does that mean? Well, it means that when I look at the design process that I showed you, I can now take bacteria as the technology on which I want to program. And again, I can have the behavior expressed in a language that can actually view this. Now I'll give you the last example before I end, and that is taking a virus instead of, and reprogramming this. It's not something that I have done, but people have been working on this, and it may sound really very, very sophisticated. But the idea here is, let's try to see if we can do something that can fight cancer. So if I have a cancer cell, I would like to program a virus so that when it enters into the cancer cell, it identifies that this is a cancer cell, and then it starts reproducing itself. So creating more and more copies of itself until the end where it gets the cancer cell to die and burst, and it releases a large number of new viruses. This is the way viruses works also in reality. But now it's do it for the cancer cell. That would be nice, because it would be something that addresses only those cells that are cancer cells. But what if we could, what would happen for the normal cells? Because that's the ones that we don't want to kill. Well, if it enters into this, it is able to detect that this is not a cancer cell, and therefore it just prohibits itself from reproducing. So it's actually dying, it's not doing anything. So it's not doing harm to the normal cells, only to the cancer cells. Now, people have shown that this is actually possible to do for certain types of cancer in the lab, in controlled environments. I think that will take some time before this would get into a more general treatment. There's a lot of problems in. I put it in a way that it sounds very easy, but these are very, very complex problems. There's so many side effects that you have to control and so on. But basically, this one could be expressed in a computer program like this, a quite simple one that just explained that if you have certain markers in the cell, then create the material that actually kills the cell. Okay, so that brings me to the end. Now, I've given you, I hope, a view on the design process and how we are thinking, and also on something that goes in a little bit into the future, what could happen. And I think I love this ancient Chinese poem that says, when the wind of change blows, some people build walls, others build windmills. So with all new technology, there is challenges, and you can think of them in the bad way or in the good way. The thing is that things are advancing so fast at the moment that we actually have to think about this. We have to be in control of how this process is going on. That brings me to the end. Thank you.